Sure.
All right, I think we're ready to go, and welcome to Persephone Ma's dissertation defense. This is the public portion of the defense where Persephone will give us a seminar and about her research, and then this will be followed by open questions by the audience. After this public portion, we will have a meeting with the committee up in 438. And we'll take a 10-minute break after, and we'll continue with the questioning then, additional questioning. Yeah. So I just want to make a few comments about Persephone's background and her PhD journey. Has been a journey. Persephone received a bachelor's degree in geophysical sciences from the University of Chicago in 2012. And after that, she worked for a supply chain company for a few years as an operations supervisor. In 2015, she took a summer class on soils at Colorado State University, which I assume sparked your interest in soils and waste management as well. She contacted me shortly after that class in 2015 about the possibility of working in my lab on a waste management project. And I encouraged her to apply to the graduate program in LAAS, and she did, and we were very fortunate to receive a CFANS Graduate Diversity Fellowship, which started in the fall of 2016. And that gave me, even though I didn't have any funding at the time, it gave me a year to figure things out. And it just so happened I was working on a greenhouse study funded by the Metropolitan Council on the effects of sewage sludge incinerator ash on both corn and lettuce. But we needed to go to the next level, and Metropolitan Council was interested in funding a field study. And so Persephone and I we wrote a grant together, submitted it to Metropolitan Council, and we were very fortunate to get funding to see her through her PhD research. Now, as I mentioned, this has been a journey for Persephone, so there's lots of twists and turns along the way. And one thing I found out that Persephone is a compulsive multitasker. So in addition to her PhD, Persephone took classes in civil engineering. She actually received a master's degree in civil engineering, I think in 2018 or somewhere around there. Somewhere around there. And other things that Persephone was involved with were involved with things like we did not have a diversity statement for our department. So she formed an ad hoc committee. She wrote a diversity statement, which is on the web for us. So she's really been very active in that area. We were very, very fortunate because the pandemic hit with all the social unrest due to George Floyd's murder. There was a lot of interest in social inequality, and Persephone helped with our roadmap, which we are still following today. And she's really helped our department from that standpoint. She's also been involved with several ballets through the Minnesota Fringe Festival. And I assume those were to give her a little break from research. And those are only the things I know that she's done. I'm sure there are other things that she's done along the way. I know I'm missing some. But with that, Persephone, the floor is yours, and we look forward to learning more about your research.
Okay, thank you very much, Carl. As he mentioned, it's been a journey, and I'm so grateful to be at this point of the journey as well. Thank you so much for being here to support me. Thank you all online as well. I try not to nerd out too much on my research, but this is my time to do so. So I will get started and let you know what I've been to besides being a cupcake and a zombie. So I am here to talk to you about sewage sludge ash as a phosphorus fertilizer. The whole story starts when my parents changed Let me see. The story starts here. This is the Metropolitan Wastewater Treatment Facility in downtown St. Paul. About 160 million gallons of wastewater, I believe, go through here. Here and many other places, they take the solids that are separated from the clean water and they burn it for energy. This produces anywhere from 1 to 5 megawatts of energy that's used within the wastewater treatment facility, but it also produces sewage sludge ash. As of now, that's landfilled. And this is the ash. Observe its qualities, pleasing color, very powdery. Every single day, they produce about 35 tons of this ash. But this ash has a lot of phosphorus. And so as a result, it also contains about 3 million pounds of phosphorus a year. Because this is landfilled, this phosphorus gets lost from any kind of recirculation. Why does this matter? So phosphorus is a mined resource. We mine it in the US, we mine it in parts of South America, but the vast majority of it is found in northwestern Africa, the continent of Africa, and China. This particular mine is a mine in Utah. And the reason why we mine phosphorus is because it is essential for all living things. It is the backbone of our DNA. It is the energy currency that we have. It makes up the energy currency that we use to have metabolic function in our bodies. And this also applies to plants. So this is soybean. The soybean on the left is grown with phosphorus. The soybean on the right was grown without phosphorus. It is this dramatic. Phosphorus in plants really determines root structure, fruiting bodies, biomass portions. And so as a result, phosphorus is regularly put on agricultural fields every year, every other year, depending on your soil type. And so the question stands is how can we actually recirculate the phosphorus that goes through a wastewater system and grow more food with it, be more productive with stuff that's actually being mined and coming out of the earth. This idea of recycling human waste is thousands of years old. With respect to sewage sludge incineration ash specifically, it's been investigated for maybe about 50 years now. Originally, when it was looked into, the metals content concentrations, the metals concern concentrations were too high. There have been changes in, at least in the US, of regulatory structures such that those concentrations have gone down. Carl actually did research, I guess, starting in the 90s throughout until the greenhouse study that he mentioned, trying to figure out whether this would be a good phosphorus source. And so the question stands, can sewage sludge ash be used as an agricultural phosphorus source? That is the background and the general impacts. I'll give you a second to just orient yourself in what I will cover. I don't have to read it for you. So get your questions ready. So I am going to start out. Questions. Does this ash provide phosphorus or not? In Europe, because they do not have mined phosphorus, they have been really interested in this question. They also have much, much stronger regulations about metals and things that you can apply to the land. So they have been doing more technical research on whether this question exists. But there are questions about whether the form of phosphorus found in this ash will actually work. And that's part of this field study. Basic question, does it work? And then how does it compare to other commercial fertilizers, which I'll talk about? Because it's a comprehensive source, because the sewage sludge is a comprehensive source, there are other things that get applied. And so we want to look at that. Are they good? Are they bad? What volumes, concentrations, loading rates, etc. And then that also includes the environmental risk portion. And so we will look at the soil chemical background as well as any impacts to the soil microbiology. And I'll talk more about that. To put you into the field study, it was done at the Rosemount Research and Outreach Center. We have an approximate climate of 7.2 Celsius, 838 millimeters. It is a Waukegan silt loam, which everyone really loves because it's very productive in Rosemount. And Rosemount is not very far away, which is really cool. We have a pretty middle 6.3 pH, decent organic matter at 3.9. It's relatively low slope, which is nice. We don't have to worry about treatments that we've applied pushing off. And we specifically picked a field that has low to medium phosphorus existing available phosphorus, because we want to make sure that any phosphorus we actually put on makes a difference. If there's way too much, it doesn't matter. So we have 4 phosphorus sources. These are the abbreviations. I will talk about them a little bit more. We've put them on at a range of rates from 0 all the way up to 80 kilograms of P per hectare. For other folks, 160 pounds of phosphate per acre. The middle 40 kilograms of P per hectare is roughly what is agronomically appropriate. So we wanted to cover that plus more just to figure out what was actually going on. I will also at some point refer to that as like the 1x rate. It's a random complete block design. We have 4 replicates in a corn and soybean rotation. So this is over 3 years. On the right side, you can see that we grew corn in 2017 on 8 replicates. Then 4 of those replicates in the next year grew corn again, whereas the other 4 grew soybean, and then we switched. The hope for this was to really have an integrated system, both that reflected crop rotations that are popular in the area, and also for us to be able to understand what does it look like when you put it on 1 year, 2 years, 2 years with a gap, different crops. And it also gave us an interesting way of looking at residual phosphorus because we only applied phosphorus to corn. So when we grew soybean, everything was what had been applied the year before. So these are our 4 phosphorus sources. I talked briefly about the ash. We got it in 2017 and 2019 from the Metropolitan Council Environmental Services. We added deionized water to about 10% for application. Tina's nodding because it's very powdery and she helped me apply it. And so we wanted to make sure that when we hand applied it and broadcast it in the spring, it would stick to the ground and not blow off. The commercial, conventional, off-the-shelf phosphorus fertilizer we compared it to is triple superphosphate. It's not super commonly used in the US anymore, but we wanted to make sure that a phosphorus-only fertilizer was a comparison point for our phosphorus-only fertilizer. We also compared it to 2 other sewage sludge waste-based products that are out on the market. One is biosolids, which also came from our lovely Twin Cities area of Blue Lake Wastewater Treatment Plant out in Shakopee. It's this particular type that we have. It comes in many, many forms. Is anaerobically digested, dried, pelletized, quality AEQ, which means it can be applied more freely and more safely than other types of biosolids. And I don't know if I mentioned this, biosolids are basically the solids portion that's separated from wastewater that can be incinerated to make sewage sludge ash. Finally, we have something called struvite, which is magnesium ammonium phosphate. It is a byproduct of the wastewater treatment process, and if not, generally what happens in most wastewater treatments, it actually coats the inside of pipes and becomes kind of annoying, and you have to do maintenance regularly. And so there's a company called Ostara who precipitates out magnesium ammonium phosphate at the beginning so that there's less of that worry, and you're also taking out 3 separate nutrients. Yeah. These were our 4 phosphorus sources. So here's some chemical information. This is the NPK agronomic values of our 4 sources. You can see that generally, except for biosolids and struvite, there is some nitrogen, but we're really focusing on the phosphorus part. For in this middle section, available P and total P, SSA is the only one that has a major discrepancy, at least by this chemical test, between available phosphorus and total phosphorus. We made the decision in this experiment to apply based on available phosphorus, because that's generally that's how phosphorus fertilizers are generally built these days. I will talk more about that because there are major implications for phosphorus loading when you apply it that way. Also provide some potassium, other things. Here are all of the other elements that we looked at. Don't look at all the numbers, but we want to bring your eye to copper and zinc. Up front, we knew that SSA had very high concentrations of copper and zinc. However, concentration isn't the only way to think about the quality or risk management of a particular land amendment. The other one is called loading rate. So based on how much phosphorus you need, how much actual material are you putting on the ground? The idea well, I didn't practice this part. The idea is basically two kinds of hot sauce. If you have a really, really hot sauce, you're going to put a very, very small amount compared to a medium hot sauce. It's the same concept here. And so you can actually see this is NPK loading as well as some other macronutrients. You end up putting significantly more amounts of phosphorus with SSA the way that we've decided to apply it in this particular experiment. However, one of the comparison points is biosolids. In our experiment, we decided to apply biosolids based on phosphorus content. That's currently not the best practices, or that's not the common usage for biosolids. Biosolids are usually applied based on nitrogen content. Nitrogen content in biosolids, depending on the biosolid, is much lower, so you end up putting a lot more product in, and therefore the loading of phosphorus based on nitrogen content, if you're applying biosolids, is significantly higher than anything that we would put on with SSA. There are many different benefits to biosolids versus SSA. SSA does not have carbon or nitrogen. That's a big difference, but for comparison's sake, in this one particular field, that is something I wanted to point out, like so. And then if we look at copper and zinc, I talked about the loading rates. Because the phosphorus content in the SSA is relatively high, you end up actually putting on more reasonable amounts of copper and zinc. Is that a bad thing or is that a good thing? Some of you might ask. So copper and zinc are unique elements in that they are plant micronutrients. They are required for healthy plant development. However, at very high concentrations, they can be damaging to plants or in the environment, depending, and so they are monitored. And so we also monitored these elements as well as all the other ones you'll see on the list. Yeah. So the overall approach to this whole project is really looking at it in three different ways. We first look at the plant biomass, the agronomic response, what is happening in the plant. Then we looked at the soil chemistry and the soil microbiology. Within these, we wanted to ask those three questions. How does the P work? How do the elements work? What are the microbiology? So we looked at corn and soybean yield and stover biomass. We also looked at P concentration and uptake in the actual plant tissue and the P concentration in the soil. That's our P question. In terms of yes, I did that. In terms of our other elements that we're keeping an eye on, we looked at, again, total elemental concentration that was in the plant after it was grown with our various types of P sources, and then the available and total elemental concentrations in the soil and so on. We also use these things called plant response simulators, which are basically resins that are buried in the soil. They come into equilibrium with the soil solution, and so it's a measure of flux. Soil solution is really where plants, for most nutrients, plants will directly get the nutrients through the soil solution, and this is supposed to be a better proxy for exactly what is available for the plants to take up. For microbial analysis, we looked at the 16S and ITS regions for bacteria, archaea, and fungi. And we sampled at a range of dates, I think 6, depending on the year, 6 times throughout the year. The analytical methods, for anyone who's curious, total elemental concentration, we used ICP-OES. Available soil elemental concentrations, we extracted with DTPA and also exchanged with ammonium acetate. And these are pictures of those PRS similar simulators that then so we would vary them, leave them for, I think it was 3 to 4 weeks, take them out, place them, and then we get a measure of flux throughout the season. Our actual results are summed over the whole growing season. I wanted to mention a couple of things about the way that we did stats. So this experimental design is something called a 4x4 factorial. We have 4 phosphorus sources and they're applied at 4 different rates. Traditionally, this is analyzed sort of as 2 categorical variables. You have how does it work? How did it work in terms of the 4 P sources and at the 4 rates? Because rate is actually like a continuous number. So we analyzed it as P source, and you're comparing the response over several rates. And so if traditionally you would have P source as many means, we actually only looked at P source and P rate as an interaction because I also mentioned that we sampled many times throughout the season. We also, because we're using the same plot and doing rotations, we did repeated measures both within one growing season and over several years to account for any correlation that might be happening with using the same plot. Okay. And I mentioned this, but it is worth mentioning it. For corn and soybean, they're both different crops, which have different, you know, physical effects on the soil, but we also made the experimental decision to only apply phosphorus when we grew corn. That is both something that actual farmers will do, or it also gives us the opportunity to look at residual. But because of that, it was we conflated basically the crops and the application, and so we analyzed that data separately. And so we will jump into the heart of things. I will show you a series of figures, and I will also explain how to read them because there's a lot of information on a lot of them. Yield and P availability, as I mentioned, is like the question. Does it work? How well does it work? This question, we're looking at SSA as a phosphorus fertilizer. So this is what a lot of people want to know. There are many different ways of thinking about what does success look like for a P fertilizer. And so we go through a number of different things. The first one is yield. That is ultimately what we were looking at. It's the highest level. And so we have the 3 years, 3 different sections. The different colors and shapes are the different phosphorus sources. Across the bottom, we have our range of application rates, and everything is looked at as a slope regression line. Over all 3 years, we had very good corn yields. I think 12,000 kilograms per hectare is like around 190 bushels per acre for those who work in that world. However, we had sort of mixed results in the first 2 years. So in the first year, we saw a response to TSP amendment and no other soil amendment. This shape is called a quadratic plateau. Is the idea that the yield will increase as long as it needs phosphorus, and then after a certain point, if there's extra phosphorus, it's not going to grow anymore. It doesn't need anymore. We saw that response in 2017. In 2018, we actually didn't see any response to a phosphorus source. That happens in the real world when you're doing field studies, but by 2019, all 4 phosphorus sources had what is this plateau, quadratic plateau response, hence only having 1 line. And so the question, just at this highest level, it does appear that at some point, all phosphorus sources, including SSA, provide an increase in yield, which is the question. However, yield is such a high level, it can be affected by so many things. There's other ways to look at whether or not SSA worked. Another is residual. So as I mentioned, soybean, when we grew it, we did not apply any additional stuff. This is only phosphorus that was applied the year. In 2018, the red line, you can actually see that it increased, though albeit small, across all of the P sources. It's average over P sources here. By 2019, if you remember, it was soybean and it was following 2 years of corn. So that means it had 2 consecutive years of P application. It has that quadratic plateau response, again, averaged over all P sources. One thing that you'll see a lot is that the question is not how does SSA necessarily differ from other P sources. The idea is that we want it to be relatively similar to the other P sources because those are commercially viable for minutes. We see that in soybean yield. But another way to look at that is P concentration. How much P phosphorus is actually going up into the plant as a average over its biomass. In corn grain, we saw an increase, again, averaged over phosphorus sources because there was no difference. This is really interesting. We saw this very early on in 2017. So when we looked at the results from the yield and nothing was responding, but we saw an increase in P concentration, we knew at least, I think that we applied the correct treatments to the right places. So that was a good sign. In corn stover, averaged over all years, you start to see a little bit of a separation in behavior amongst the P sources. This doesn't, for those, we used a p-value of 0.05. This was 0.07, but the trends are starting. In P concentration in soybean, again, by that residual year, whenever it's taking up concentration, the differences in P source dissolve away. They all increased in every year, a little bit more in the soybean grain, the top blue line, in the second year, which was following 2 consecutive years. Another way to think about nutrient use efficiency is how much did you apply? How much would the plant have behaved without you putting anything down? And then how much did your actual action change how much yield there is? So ergonomic efficiency is the yield minus whatever yield you got with the control, because that would have existed anyways, divided by the actual phosphorus that you applied in the field. Agronomic efficiency based on yield very much follows what we saw in the yield graph. In the first year, TSP is higher, generally higher than the other P sources. But by 2018 and 2019, they start to even out. If you do apply the same logic to uptake, which is literally how much phosphorus product is in the plant, we start to see that things even out a little bit more. This difference between yield and uptake, we don't have a great answer for, but I think the main takeaway is that by 2018 and 2019, all of the phosphorus sources are behaving similarly. We will get more into why that might be. But this is another way to wrap your head around exactly whether this was successful or not successful. Olsen-P is a measure of available soil phosphorus. So we've already looked at the plant. We know what's going in there, why it's going in there. But how much phosphorus is in the soil? That's another question, because that's where the plant gets it from. Olsen-P is a measure of that. Before I get into this, which is a lot of lines and a lot of letters, I will say that Bray-P and Olsen-P are the traditional soil tests. The typical soil test for our soil would be Bray, but existing literature has found that because SSA is such a basic P source, that the extractant used in the Bray-P soil test has a tendency to exaggerate how much is actually available with phosphorus with SSA. For that reason, we're going with Olsen-P to give a more realistic understanding of what is actually available. Since soil P tests are extractants, they're proxies for what we may or may not think that the root exudates in the soil system would be doing. So on this graph, all the different colors, P sources again, we have the rates across the bottom and the years top to bottom. This is a time series of our tests, our soil tests throughout a given growing season. To simplify this, I'm just going to pull out TSP and SSA, because that's the one we're really interested in versus our commercial comparison. One of the trends that you'll see is early in the season, mid-June to mid-season, TSP consistently has a stronger response in Olsen-P than SSA. However, by the end of the season, the difference starts to taper out. This is in line with what we would expect. TSP is specifically billed as 100% water, 100% available phosphorus source. So when you apply it, plants should be able to take it up. There are oftentimes when you apply phosphorus to the soil, after a certain amount of time, that phosphorus binds to the slump matrix, and that may be what's happening as you see throughout the growing season. It becomes less available being the Olsen-P. However, I will point out that each of these lines has a slope that's greater than 0. So in every single situation, when you're applying SSA, when you're applying TSP, it increases the available P in the soil. If we wanted to compare SSA to the other residual-based products, it gets a little bit more complicated. We actually see struvite having this early season peak in 2018 and 2019. But then again, mostly by the end of the season, all of them behave relatively similarly. I think it is interesting to mention at this point, I won't talk too much about struvite, but struvite is billed as a slow release phosphorus fertilizer. The forms of the magnesium ammonium phosphate supposedly is not as available as TSP as we saw on the previous graph. And we can sort of see that here. It has a much stronger response in 2018 or 2019, though it still does some things in 2017. Another measure of success, perhaps, is this PRS-P. How much of that phosphorus is actually getting into soil solution so it's available to be taken up by plants? Generally speaking, 2017, 2018, there are not significant differences, though you can start to see the trend of SSA having a lesser response than the other phosphorus sources. When you see these letters on a graph, I should have mentioned that earlier. The letters, different letters mean that they are statistically significantly different from each other. And so A, the struvite response, is different than B, which is the biosolids response. However, from an SSA perspective, you can start to see that it's moving towards the middle. This slow release behavior that I've mentioned with either the residual response in soybean or that we saw previously in Olsen-P and PRS-P, there's a number of different things pointing to that part. Ultimately, though, again, as I mentioned in Olsen-P, all of these lines go up. That means when you apply SSA, there is something happening. It is increasing the amount of phosphorus in the soil system and therefore actually available, which is nice. One thing that I alluded to, which now I will actually talk about, is the difference between total soil P, total phosphorus, and available phosphorus. Phosphorus comes in many different forms, and some forms are available to plants, some forms are not, some forms change in the soil, some do not. This is total phosphorus. No matter what species, what are you putting on? And as I mentioned, we are putting on basically twice as much total phosphorus with SSA than any other P source. That was a choice that we made in this experiment, and you can start to see it. This is again 3 different years, beginning of the season, end of the season. We start with no differences. By the end of 2019, you start to see the total phosphorus concentrations pulling away from the other phosphorus sources. That is something to be aware of. Phosphorus is responsible for environmental degradation if it's in very, very high or high concentrations in particular species that can be damaging. This is something worth keeping an eye on. At these particular concentrations, they're not enormously worrying, but it is something to consider when you're advising someone on how often you're going to put SSA on. Is it every year, every other year? What rates, where? One thing I also wanted to point out too was that between 2017, the end of the 2017 season and the beginning of 2018 season, you see all the differences in P source disappear. Phosphorus doesn't leach the way that nitrogen does, and so that was a question that Carla and I had. Where is the phosphorus going and why is it changing like that? One thing is that in between the end of season and the beginning of the next season, we disked in the top 30 centimeters of the soil, basically diluting the treatments that we put on and digging it deeper into the soil. Because of that, it probably diluted or diminished some phosphorus or some phosphorus source responses that we saw. Therefore, between end of 2018 and the beginning of 2019, total phosphorus was really high and then it kind of went away. That is likely what's going on. Ultimately, for the phosphorus question, we have proof both in the yield, concentration, soil solution, or soil chemistry that phosphorus is being released from SSA. That is great in terms of using it as P source. There is a lot of literature that kind of says, SSA, it's an incinerated project, it's highly inert. The species in phosphorus in SSA cannot be used by plants. This is one of the very few field studies that are done, and we actually have proof that that's not quite true, though it could depend on a number of other things. But in terms of this particular field study, that's the answer to our first question. So the next question is, what else is coming with this SSA that we applied? There's always questions with human byproducts of metals or contaminants. Because it's incinerated, for the most part, we don't have to worry about antibiotics, pathogens, things like that. We're really concerned about the metals or metalloid concentrations, so things like lead and mercury and all that stuff. That giant table that I mentioned at the beginning, those are all of the elements we looked at. I'm going to talk about the ones where we actually saw a response and circle back to the ones that people might be concerned about, but we didn't actually see anything. So I talked about copper. This is just like Olsen-P, DTPA copper is available, kind of like available copper. And then PRS copper is the soil solution. You see that the letters A, B, and C, they start to separate. Biosolids and ash lead to a significant increase in available either DTPA or PRS copper compared to the other sources. The interesting thing here is that we have a bit of a difference between DTPA copper and between biosolids and struvite and SSA, whereas in the soil solution, biosolids seems to be like way available. A lot of this, again, has to do with the fact that probably it's a totally different composition. You're applying different amounts. And it's, for me, it was more just a interesting idea that we are looking at different things. You're looking at a snapshot in DTPA, a snapshot of what available copper says it is there versus moving flux of what's in the soil solution, what actually transfers from the soil into something that is plant in a form that plants can take up. We also saw an increase in total copper. So at the beginning of the season, not so much of a difference because of that disking effect that I told you about. By the end of the season, both biosolids and SSA resulted in significant increases in total copper. At these rates, it doesn't get anywhere close to environmental damaging levels of copper. This is very much in the plant micronutrient concentration range. Same thing with zinc. We see the same response. So I'm just going to let this be here because it's the same as what I just said. However, the big difference with zinc is we didn't see an increase in total zinc, but we did see it actually translate into the crop body. So in corn grain, corn stover, and soybean grain, the application of biosolids and ash resulted in higher concentrations in these three than the other resources, which is great because this is actually evidence that biosolids, which we already knew, but SSA provides zinc, which is a plant nutrient. It's a comprehensive source right here in action. Other responses were in exchangeable potassium and sodium. So these are sort of two opposite sides. Potassium is another macronutrient that plants need. We actually applied potassium to our fields to make sure it was even. But even with that, we saw a response that SSA provides potassium. It's getting into the soil. It's being taken up. It's in a form that conventionally seems to be available for plants. Sodium, on the other hand, is more concerning. It's can have pretty damaging effects to soil if you're at really high concentrations. There is an increase with biosolids and SSA. Again, one of the big trends is that this increase, and in the scale of 3 years of our experimental field trials, doesn't have significant biological implications. However, it's something that if you were to apply SSA over and over again over several years, is something to keep an eye on. So we get to elements that people really want to know about. I will say that mercury is the only one that we saw a response. Lead, arsenic, cadmium, and chromium, which have been found to increase in other past SSA field studies, did not increase in our particular scenario. Total mercury, on the other hand, did. And we know that there is mercury in this ash. There's just the pure idea that we know it's in the ash, putting it in the soil, therefore it is increasing. It is increasing to a somewhat statistically significant amount, but I will also point out that the y-axis is in micrograms per kilogram, which, again, the biological significance of a 4 to 7 increase in micrograms per kilogram is relatively small, but again, something that should be followed over multiple year applications. So we went through a couple of elements, some good, some bad. It is a comprehensive source, I'm just gonna keep saying that. But the question is, if we apply ash and it works as a phosphorus source and it doesn't have major detrimental effects because of other elements that you're applying, that's good. But if it destroys all the microbes in the soil, which are an integral part of soil fertility and life and all that stuff, then that's really bad. So we wanted to make sure we weren't doing that. The first step that we actually did was a proof of concept incubation. We just took soil, put ash at 0 to 1,000 times what would be agronomically appropriate for a field. just to get some sense of, will we see anything or will we not? We didn't really see anything. So this is called a PCOA graph. The general idea is that the clusters, they are very far apart from each other, which I'll show you later. Then they are significantly different from each other. These overlap a lot. There's a difference in shape, but generally, even if you were applying 1,000 times the amount that you should, we didn't see any response, any changes in the microbial community in an incubation. And incubations and field studies are very different. And so we did continue on to the field study and do metagenomic analysis at that point. One way of asking whether or not there are significant changes in the metagenomics is something called relative abundance. This is a visual indication of whether or not there are differences in, if you didn't do anything versus when you applied something. So all of the treatments for the 4 different P sources are down here at the 1x rate and at the 2x rate, not at the 1,000x rate. We did not do that in the field. There's some variation in terms of trends based on P source or anything else. There's not anything significant. This is for bacteria. Same for archaea. Nitrososphoraceae is highly dominant in this particular, that family. In fungi, we do start to see major variation. We were actually really hoping to see something in fungi. Fungi is a lot more reactive, and it also has a very tight connection with phosphorus use in the soil. So since we're applying phosphorus, we thought maybe fungi would do something. However, looking at this visually is kind of like, okay, how do we parse this all out? And so, similar to that PCOA graph that I showed you previously, this is fungi, based on phosphorus source. All of the data points are on top of each other. The general ellipses were overlapping. There's no difference between phosphorus sources with that fungi. And this is exactly what it looks like for bacteria and archaea as well. If in case you were curious about what a PCOA plot looks like when there are differences, we did find differences which match literature, just that different years and different crops will have significant impacts on your microbes. This is also fungi, but similarly in bacteria, a little less so in archaea, excuse me, we see these separations start to move. Most likely, these are much more dramatic factors compared to the minute amounts of phosphorus that we applied. And so we did see something. Again, it's a nice indication that we're actually doing something correct, but we didn't see anything with SSA. And this was really a study of like no differences or good differences. And that was what we liked to see. So I've sort of covered the P question, elements, metagenomic information. We have proof that whatever we're applying isn't going to kill everything. Always nice. And so what we found is SSA increases available and total phosphorus in soil and appears to provide a slow release form of P for plant uptake. This SSA provides copper, zinc, and potentially other elements at appropriate concentrations for plant nutrition. Currently, SSA amendment leads to increases in exchangeable sodium and total mercury in soil, but does not result in any increases in biomass that we saw. SSA applied at agronomic rates, and even at 1,000 times their agronomic rates apparently, does not affect microbial communities any differently than commercial P sources. Because this is one of the first field studies, there's much more to look at, and it runs counter to a lot of the research or the thought process, at least, that European studies have found about SSA and how well or whether it works as a phosphorus source. And so the next question I have, really, if I had the time, is the characterization of P species in SSA and exploration into correlations with fertilizer capacity. So we know that whatever is in this SSA leads to yield increases, but I don't know what it actually looks like in the SSA. What are the species of the phosphorus? Which ones are actually being taken out of the plants? Can that be manipulated? Questions like that. We've also seen that, like, we used citrate soluble phosphorus levels, that available phosphorus, to apply. It is not really a direct it eventually led to yield, but it doesn't seem to be a good indication of fertilizing capacity like it is for commercial sources like TSP. There have also been existing meta-analyses saying that residual phosphorus sources, traditional analytical tests of availability or fertilizing capacity are not really quite accurate. And so that should really be explored if we want to have the system to support common usage. As I mentioned, investigation of concentrations, fate, and transport of contaminants. Oh, I didn't mention this one, sorry. Contaminants of emerging concern. So bring on the PFAS questions. It was not in the scope of the study. I was lucky, I think, to start it before that conversation started. We are following the research, but it was not in this study. As I mentioned, product development, it's very powdery. The questions of how to make it easy for people to use is still an existing one. On-farm testing under realistic growing conditions in a variety of soil types and crops. We did corn and soybean, but what about tomatoes and lettuce in a field, or wheat? Who knows? We also had a very nice soil to work with, but what about sandy soils? What about different climate schemes? Periodic monitoring of soil P tests and soil concentrations of elements of concern. That total P problem, that P loading issue needs to be addressed. And also built into the development of best practices for SSA application. It include guidelines for the testing that I mentioned, soil testing, and SSA application. When do you apply it? How often? At what rates? Where? In what forms? All those lovely things that everyone else works on. So I would like to acknowledge Metropolitan Council, Environmental Services, all the folks there who financially and academically supported this work, the CFAMs, UMN Graduate School, and also the Baker Family Foundation I probably could have done some of it without them. It would be a lot less enjoyable without financial backing, so I'm really grateful to them for that. I want to give a special thanks to Carl, my advisor, who has stuck with me for these long 6 years, as well as my defense committee, Dan, Sebastian, Brandy. I'm looking forward to additional conversation. I also want to thank the Rosen Lab, the SWAC team, all the people who have made that possible. Rosemount field crew, research analytical lab, the SWAC field crew, and Aaron helped with stats. As you noticed, we had a very complicated experimental design, which made stats really fun. I feel good about stats now. These are all people who made sure I got to this finish line. There are also people who made sure that my body and brain in one sort of piece got to the finish line. My parents and those who I come from were a huge part of that. My colleagues, collaborators, friends, everywhere across the globe were so important for this whole process, and Dylan as well, supporting me and moving to the lovely state of Minnesota with me. Yeah, I could not have done it with so many people, and I am so grateful to be here and open to your questions.
Minutes for questions, and so yeah. John.
Persephone, probably the major environmental question for phosphorus is contamination of surface runoff. I know your site wasn't really conducive to those kind of measurements, but based on the various things that you did measure, do you expect that there would be any effect on the surface runoff losses for an SSA versus phosphorus?
I think just off the cuff, I would say no. I think additional analysis and speciation of the phosphorus in the SSA will help answer that question. For the most part, the assumption is that it is in forms that is less water soluble, which is really the issue with TSP. SSA does not appear to be that water soluble, and also it has this very, very delayed response, which implies to me that it's in forms that it's less available in the spectrum of availability for phosphorus. One thing I didn't really talk about is though I alluded to, is there's this paradigm that when you apply phosphorus, when you buy a fertilizer, you should be able to put it down and plants should take it up immediately. Because it's water soluble, it also increases the likelihood of soil solution runoff and the loss of phosphorus two places where it shouldn't be. That's always, you want phosphorus some places and not other places. And so products like struvite, that's what they're being billed as, reducing the amount of water soluble phosphorus so you are less likely to lose it. And SSA could maybe also fit into that style and changing the paradigm of how you apply phosphorus fertilizers. Oh, sorry. Yes. Thank you for the reminder. The question was broadly whether we expected to see effects in phosphorus runoff due to SSA application, right? Roughly. Brandy.
I apologize if I missed this, but did any of your treatments have the sludge phosphorus with a conventional type? And I was wondering what you thought about everything from runoff to availability to anything else. If it was, let's say, some ratio of kind of conventional phosphorus with the recycling sludge source.
Yeah. So for those online, the question was whether we mixed the sludge ash or the sludge with the conventional, more available phosphorus, and what implications that treatment might have. So we didn't. We had just TSP, just biosolids, just ash, and just struvite. However, there are other studies that have done that, and based on this balance of meeting crop needs early in the season and also just the long-term need for phosphorus, that would be a sort of best practice or some kind of technique for using the ash that might be beneficial to explore. So if you remember back to the comparison between SSA and TSP, there was a big peak at the beginning of the season, and then it sort of disappeared from there. An approach could be having some TSP so that you meet that early season need, and then have the SSA be a longer term need, either for later in the season or winter, something like that.
Great, thanks.
Mm-hmm. That's right, Nick. And then, yeah.
So you evaluated both biosolids and SSA.
Mhm.
The focus on SSA is because here, that's just what we do, right? Like, so in St. Paul, we're incinerating these materials. So from your research, do you, like, there's a reason that we're doing that, obviously, on the reason why. But if you had to recommend just from a reuse perspective, is the incinerated stuff better?
Oh, how many people do I want to use? So that's who didn't hear the question. But I compared both biosolids and basically the solids ash, the sewage sludge ash in this experiment. Nick is asking the hard question of which one's better. And yes, the reason, I mean, practically speaking, the reason is because the reason they incinerate here is because the place in which wastewater treatment happens is a very limited physical area. And so it's a lot easier to incinerate than do biosolids management. There's like a lot more volume, a lot more to do lagoons, shipping, all of that. It was also, there were also concerns about smells, odors, biosolids that you don't have with incineration. But as I mentioned before, incineration ash does not have carbon or nitrogen. This does. There are folks in the world that think that it is a sin to incinerate biosolids. I did not make the decision to incinerate biosolids. I just happened to be profiting vaguely. Profiting might be an extreme word, from that decision. It really, I'm going to give the unfortunate scientist answer. It depends. It depends on your ash. There's wide variation between ashes. That's what I found. We happen to have very good sewage sludge ash in the Twin Cities. So yay us. It's high. All of you are responsible. So it's very high in phosphorus and it's very low in metals. But depending on where it comes from, if you have high amounts of industry, if you're in a country that doesn't regulate as well as the US, that has a huge impact on the characteristics. So it depends on your ash. It depends on how close you are to agronomic land, because it's a lot easier to ship incinerated material than biosolids, even if they are dry and pelletized. It has to be sort of a decision based in the space that you have the product, I think, unfortunately. I think they both have their place. some places more than others. Maybe the PFAS question will answer. I don't know. Yeah, sorry, somewhat sciencey answer. Oh dear.
It's controversial.
No, I like it. Go. Yeah.
Talk all day.
Yeah. There's a meeting I think that you could go to. John, I think you had a question.
Yeah, so are there any current rules or regulations that prohibit the use of these products for if not, then what are the barriers that are keeping us from using?
Yeah, so there is no specific regulatory policy with sewage sludge incinerator ash. There is national policy on biosolids application, and that was what we broadly generally used to frame how we thought about questions that we ask for using SSA as a land amendment. The question, it sort of lives in this purgatory of like, are you going to try to use SSA without regulation and see if they catch up, or are you going to wait for the regulation to exist before you apply it? I don't know the answer to that. It is actually being land applied in a few places in the US, in parts of New Hampshire and Ohio. They are mixing it with soil amendments because it's so powdery. It actually basically lightens the soil, but they're using it more for landscaping as opposed to agricultural usage. So I don't know. I think it's like a race for whoever moves first kind of question. Oh, sorry. The question was whether, which was maybe implied by my answer, but whether there was any regulatory scheme that oversaw SSA land application.
Well, I do think you'd have to contact MPCA about land application before doing it. I think Met Council is in conversation with
Oh,
MPCA
yes.
about it. I don't think it
Yeah,
can.
that was more like a thing.
No,
Oh, okay.
even with the ash, they're gonna have to.
Well, I stand corrected.
Yeah.
There is no regulatory,
There's
there's no
something
specific
yeah,
law.
but I
that
think
says-
there's got to be some contact with
Yeah.
MPCA before you can land apply it.
Yes, for those who couldn't hear it, and Med Council who produces and owns this ash is in conversation with MPCA about it. So however that works. Thank you.
There are no current regulations.
And the biosolids regulations really do not apply to SSA. They're totally different products. Yes, David. Okay.
Is there anything enough measurements to do already without looking at total and soluble organic P? Do you have any idea how those fractions might differ in your treatments?
So the question for anyone who didn't answer is the organic fraction of phosphorus, how that would differ between treatments. I mean, just off the cuff, biosolids has an organic portion of phosphorus, and so therefore that would likely lead to an increase in organic phosphorus in the soil solution, whereas SSA is totally inorganic. Everything organic is pretty much burned off. It's like 0.5% carbon. For struvite and TSP, I think I would believe that they would behave similarly to SSA and that there's pretty much no organic friction, and it doesn't add any organic matter that might bind the phosphorus. So any binding would happen in the soil itself. It would probably generally increase the organic phosphorus a little bit because you're applying phosphorus and it sticks to things. But yeah, I think that would be my guess. We did not look at the organic portion of very much at all. Tried to keep it somewhat limited. Thank you.